A deep diver. Ever wish we had, like, these tiny customizable tools,
Yeah.
tools that could track down anything, a virus, maybe a toxin, or how about even a rogue cancer cell?
Yeah, it's wild, right? Scientists are already on it. And
Huh.
they're called aptamers.
Aptamer?
This July 2024 research article, it dives into, how we even find these microscopic wonders.
Okay, so we're talking about a scientific journal here. Very cool. But by the end of this deep dive, we'll both sound like, we're in the lab, right?
Absolutely. Think of aptamers kind of like molecular Sherlock Holmes, so to speak.
Hmm.
They're sniffing out specific molecules with amazing precision. It's incredible.
Oh, right.
They're like antibodies, but like on a whole new level.
Okay.
Smaller, cheaper to produce. Right. And you can design them to bind to like a way wider range of targets.
All right, you've officially got my attention. So how do we find these aptamers? Is it like searching for like a specific grain of sand on the beach?
That's where SELEX comes in.
SELEX.
Short for systematic evolution of ligands by exponential enrichment.
It's catchy.
So imagine a giant library, right, of DNA or RNA strands. Each one is a potential aptamer.
Okay.
Scientists will mix this library with that target molecule, the one they're interested in.
Okay, so we're throwing like a target into this massive molecular library. What happens next?
So it's like a giant game of molecular matchmaking, I like to think.
Mm-hmm.
Only the aptamers that successfully lash onto that target, those are the ones that get to move on to the next round.
Oh, so it's like
Or rest.
survival of the fittest, but on a molecular scale.
Exactly, yeah.
You like it.
So scientists take those winners, amplify them, which basically means they create multiple copies, and then they repeat that process.
Okay.
The mixing, separating, amplifying,
Mm-hmm.
And after several rounds of this, you're left with a very select group of aptamers, and these have a super strong affinity for that target.
This SELEX process, it's pretty ingenious. It's like, letting nature do the heavy lifting of finding that perfect molecular match. Yeah, it's incredibly powerful. And, scientists always trying to improve things. They've been working on ways to make it even faster and more efficient.
Faster. I'm listening. How do you speed up something that already happens on such a small level, a microscopic level?
Well, there are a few different approaches. One is called CE-SELEX.
C-SELEX. Okay, break that down for me. What is that?
So that stands for capillary electrophoresis, SELEX.
Yep.
And imagine separating a different colors in a lab using a special gel, and that's kind of similar to what's happening here. But instead of colors, we're separating aptamers based on their size and how quickly they move through this gel-like medium.
Okay.
It lets researchers identify the best binders much faster than, traditional methods.
So it's like giving each aptamer like its own tiny racetrack, and the fastest ones, those are the winners.
That's a great way to put it, yeah. And it gets even more interesting. There's another technique called FluMag-SELEX.
FluMag-SELEX. I feel like we're inventing words now.
Laughs. It does sound kind of futuristic, right?
It does.
But it's actually a very clever technique that uses fluorescence and magnetic beads.
Okay, you're gonna have to elaborate on that one. I'm picturing like tiny magnets and glow sticks.
Yeah.
But I'm pretty sure that's not how it works.
You're on the right track with the glow sticks. So we attach like a fluorescent tag to each aptamer, and that lets us easily track them.
Okay.
And the magnetic beads, those act like tiny magnets, attracting and separating the aptamers that have bound to the target.
So it's about making that selection process, faster and more visible.
Precisely, yeah. And then there's in silico SELEX.
In silico SELEX.
FluMag, which takes the whole process digital.
Digital SELEX, as in doing this all on a computer.
You got it. Scientists are using, these sophisticated computer simulations to test millions, even billions of aptamer sequences virtually.
Wow.
And they don't even have to, set foot in a wet lab.
Wow, that's amazing. We've gone from, giant molecular libraries to computer simulations in just a few minutes. What's next? Are we going to be evolving aptamers in space?
You never know. But in all seriousness, these advancements in aptamer selection, they're already leading to some really incredible real-world applications.
Okay, I'm on the edge of my seat here. What can aptamers actually do? Give me some examples.
So aptamers, they're already making waves in the field of diagnostics.
So earlier detection of diseases, that's huge, especially for something like cancer, where that early intervention can be so crucial.
Exactly. Imagine, imagine a world where instead of invasive biopsies, you could have like a simple, accurate test. And, using aptamers to detect those cancer cells that are just circulating in the bloodstream. That's the future we're talking about.
Wow, that would be revolutionary.
Yeah.
But it sounds like aptamers can do a lot more than just diagnose, right? You mentioned something about like therapeutic potential.
Absolutely. Imagine designing aptamers. Aptamers that could deliver medication directly to a specific target in the body, right? Minimizing side effects and really maximizing effectiveness. It's like having like a microscopic guided missile for medicine.
That would be incredible. What kind of diseases are we talking about, like treating with this kind of precision?
Well, researchers are exploring aptamer-based therapies for everything. I mean, from age-related macular degeneration, a leading cause of vision loss, to bacterial infections and even cancer.
Okay, so aptamers are showing promise in diagnostics and therapeutics, but what about, other areas? Are there any, I don't know, unexpected places where aptamers are making difference?
One area that might surprise you is food safety.
Food safety. Now that's something I would have expected.
Yeah.
How do aptamers play a role in keeping our food safe to eat?
Well, remember how we talked about aptamers being able to detect even the tiniest traces of specific molecules? Well, that includes, harmful substances like pesticides, mycotoxins. These are things that can contaminate our food supply.
So aptamers can act like microscopic food inspectors,
Yeah.
ensuring that what we're eating is safe. That's amazing. But, with all this incredible potential, there must be some challenges too, right? I mean, it can't all be smooth sailing.
You're right. There are definitely hurdles to overcome. One major challenge is that aptamers can be broken down by the body relatively quickly, which, can limit their effectiveness, especially for therapies.
So it's like we're sending our molecular detectives into like a hostile environment where they might get ambushed before they can complete their mission.
A very apt analogy. So to address this, scientists are developing ways to make aptamers more stable. For example, they're experimenting with, attaching a protective molecule. It's called PEG to the aptamer, and it acts like a shield, so it makes it harder for the body to break down.
That's clever.
Yeah.
Like giving our molecular detectives, a bulletproof vest. But what about, accidentally hitting the wrong target? How do we make sure that aptamers are only binding to the molecules they're supposed to?
That's another really important consideration. And researchers are working on improving, aptamer specificity. One strategy involves like exposing the aptamers to a variety of molecules, including some that are similar to the target but not an exact match.
So it's like a molecular training course for our aptamers,
Hmm.
helping them to distinguish between the real target and any decoys.
Precisely. This helps to weed out any aptamers that might be prone to like off-target binding,
Hmm.
ensuring that only the most accurate and specific aptamers are selected.
It's fascinating how scientists are constantly refining and improving this technology. It's like a never-ending quest for that perfect aptamer.
It really is. And, with each advancement, we unlock new possibilities for, diagnosing, treating, and even preventing diseases. Take pegaptanib, for example.
Tigoptanib, what's that?
It's actually the first aptamer-based drug approved by the FDA for the treatment of age-related macular degeneration, that eye disease we were talking about earlier.
Wait, so they're already being used to treat a leading cause of vision loss? That's incredible. Are there other aptamer-based therapies in development?
Absolutely. Yeah. There are a number of them in various stages of clinical trials. They're targeting everything from cancer to infectious diseases. Researchers are even exploring ways to use aptamers to deliver drugs, not just to specific cells, but to specific compartments within those cells.
Wow, that's incredible. It's like having a microscopic delivery service that can bring medicine exactly where it needs to go within the body.
It's a rapidly evolving field with incredible potential. Another area that has researchers particularly excited is the fight against, antibiotic-resistant bacteria.
Superbugs. How can aptamers help us win the war against those?
Well, unlike traditional antibiotics, which often target broad classes of bacteria, aptamers, they can be designed to target very specific strains, including those that have developed resistance to, conventional treatments.
So aptamers could offer a more targeted approach, like reducing the risk of wiping out, the beneficial bacteria,
Yeah.
and potentially even slowing down the development of resistance.
Exactly. It's a game changer in the making.
It's amazing to think that these tiny molecules, discovered just a few decades ago, are already having such a profound impact on medicine. It really highlights the power of scientific curiosity and innovation. But with all these, amazing applications, I can't help but wonder if there are any, limitations to what aptamers can do. Are there any targets that are simply too big or too complex for them to handle?
That's a great question. While aptamers are incredibly versatile, there are some limitations to consider. For example, they are typically smaller than antibodies. which can make it challenging to target certain, large or complex molecules effectively.
Oh. So are there any workarounds for that?
Scientists are exploring ways to overcome this by creating what are called, multivalent aptamers, which
Right.
are essentially, multiple aptamers linked together, like a team all working together to secure a larger target.
Ah, so instead of one tiny detective, we have a whole squad of them working together.
Precisely. It's a very dynamic field, and researchers are constantly developing new strategies to push the boundaries of, what's possible with aptamers.
Well, it's clear that aptainers have a lot to offer the world, and I'm excited to see what the future holds for this groundbreaking technology. But before we get too caught up in the possibilities, let's circle back to some of the challenges we discussed earlier. You mentioned that, aptamers can be degraded by enzymes in the body. Are there any other strategies, besides pegylation being explored to address this issue? So pegylation is like giving our aptamer detectives a protective shield. Are there other ways to make them more durable out in the field?
Oh, absolutely. Researchers are exploring, a whole arsenal of modifications, like tweaking their chemical structure, or
Mhm.
even mimicking those naturally occurring molecules that are just, more resistant to degradation.
Oh, so it's like giving our aptamers like specialized training
Right.
to survive in even the toughest environments.
Exactly, yeah. And as we develop these new strategies, we're also getting better at, predicting and preventing those potential off-target effects.
How so?
Well, remember how we talked about using, counter-selects to improve aptamer specificity?
Right.
Scientists are like taking that concept even further. They're using sophisticated computer modeling now to actually predict how those aptamers will interact with different molecules in the body.
So it's like giving our tamers like a virtual map of the body.
Right.
To help them, navigate and avoid any wrong turns.
Precisely. These computational tools they're becoming so powerful, allowing us to design aptamers with greater precision and safety.
It's incredible how technology is shaping the future of medicine, even at like the molecular level. This whole deep dive into the world of aptamers it's been really eye-opening.
Oh, it's such a fascinating field, and it's only just the beginning, We've covered a lot of ground today, from, the basics of aptamer selection to some of the most, cutting edge applications, but there's one more thought I'd like to leave you with.
I'm all ears.
We've talked about aptamers that can target specific molecules, cells, even viruses, but what if? What if we could evolve aptamers that target processes?
Processes, what do you mean?
Imagine aptamers that could disrupt the formation of a blood clot, right? Or
Uh-huh.
or prevent a cancerous cell from replicating. Aptamers that could intervene in the very mechanisms of disease.
Whoa, that's next level stuff.
Yeah.
It sounds like science fiction, but is that even within the realm of possibility?
Well, that's the beauty of scientific exploration, right? We're
Right.
we're always pushing those boundaries of what we think is possible. And with the incredible versatility and specificity of aptamers, I wouldn't rule anything out. As we continue to unlock their secrets, who knows what amazing applications we'll discover, right?
I don't know about you, deep diver, but I am feeling incredibly hopeful about the future of medicine.
Mm-hmm.
It's been a privilege to learn alongside you today. Until next time, keep those questions coming and never stop exploring.
Until next time.